Okay, so I can introduce the last speaker. Last speaker is Nigel Rogasch. Sorry, I have to take it. So, he is from the University of Adelaide. His research concentrates on combining TMS with neuroimaging techniques like EEG and fMRI to understand the role of excitability, excitatory and inhibitory mechanism, brain organization and plasticity in healthy and unhealthy humans. He is also a main contributor to the BEP proposal to create a NIBS structure for non-invasive brain stimulation, which is the project that he's going to present today. Thank you.
Thank you very much, Marta, and thank you so much for organising this symposium. I think it's a really fantastic initiative, so well done. So today I'm going to talk a little bit about an extension proposal that we have for the brain imaging data structure to try and include data related to non-invasive brain stimulation. So Anthony's already given us a really good example of this, but reproducibility is a major issue that's facing all of us at the moment. This is just another example that was a large-scale study looking at a number of different psychological studies that found that only about a third of these major studies actually replicated, which is I guess on one level quite concerning. And it's concerning for a number of reasons, but I think one which is quite worrying is the drop in trust in scientists that we're sort of seeing across society at the moment. So these reproducibility issues are something that we really need to tackle. There's been a number of different ways that have been proposed to do this, but one of the main ways, I guess, is through using open science practices. And this can include various different approaches, such as sharing data to increase transparency, sharing the code and analysis pipelines, and also making sure that the papers that we publish are accessible to everyone. But today I'm going to be focusing mainly on the data sharing aspect of the open access pipeline. So data sharing is not only important for reproducibility, but there's many other really good reasons to share data. One is that it makes that data accessible to others for further use, so it really maximises the benefit that we get of all of our hard work. And some even argue that there are ethical requirements on our behalf to try and maximise the use of this data that we collect. So there have been some principles that have been put forward about how to share your data well, and one of these is the FAIR principle. So this involves making your data findable, so giving it unique identifiers, making it accessible so that people can get to that data, making it interoperable, meaning that it can be used across multiple different software platforms, and making it reusable, so making sure that the licensing that you include with your data is such that it can actually be reused by other people. So the neuroimaging community has actually been onto this for quite a while, and there's been a number of different data sharing proposals that have been put forward. But one that's really gained traction is the brain imaging data structure, or BIDS, and I'm sure many of you already know about BIDS, but it was a structure that was put forward by Chris Gorgolewski and Russ Poldrack nearly 10 years ago now. At its heart, there are three core aspects to the brain imaging data structure. So this provides an agreed way of having your file name and folder organisations. There are some standard file formats that are recommended for different neuroimaging types, and also for additional data that goes alongside. So they're using things like text files, tabulated text files. And then there's also these additional JSON sidecar files which can be used to include additional metadata about the data that you're sharing. These JSON sidecars are important because the JSON file format is both machine readable, so it can be read easily by computers, but is also quite easily readable by humans as well. So it sort of makes that information quite digestible. So BIDS started out really in MRI, but over the last decade has really expanded out into a number of different modalities, including MEG, EEG, PET, and near-infrared spectroscopy, just to name a few. And it really is achieving its goals. So this is some data showing how many shared data sets and participants are now available on the Open Neuro platform, which takes BIDS formatted data. And so we're up to nearly, I think now over 1,000 cumulative data sets from over 35,000 individual participants, which is quite a remarkable effort from the neuroimaging community. However, the field that myself and several of us work in non-invasive brain stimulation, doesn't actually have currently a way of standardising the way that we share the data that we generate that's related to these techniques. So Marta's already talked a little bit about transcranial magnetic stimulation, but non-invasive brain stimulation covers a whole range of different techniques, including transcranial electrical stimulation and transcranial focused ultrasound stimulation, which would be the main ones. So, as part of the T4TE project, Marta and her colleagues in Italy decided to put forward an extension proposal for BIDS to include ways for sharing non-invasive brain stimulation data. So this is being led by Giacomo Bertazzoli and Marta and a few others. And as with all BIDS applications, this is really a community-run and led program. So there's quite a number of different contributors from across the brain stimulation community and the BIDS community that are working on this extension proposal. So when we think about what kind of things we actually want to have access to, or the information that we generate with NIBS experiments that would be of interest, there are things like what devices that we use to stimulate the brain, so what coils, transducers, electrodes, and the devices themselves, where those stimulating coils, electrodes, transducers were placed in relation to the head, when the stimulation took place, and how that stimulation was delivered. So things like the intensity of the stimulation, as Marta was just talking about in the case of TMS, the pulse shape, so a bunch of different things that are of interest. So NIBS does present a little bit of a challenge in that, in and of itself, it's not actually a data recording modality. So when we provide stimulation, we typically need to use some other kind of neuroimaging modality to actually capture the response of the brain. And this is typically done in two ways. So the first is using what's called online stimulation. So this is when we provide stimulation while we're actually using another neuroimaging modality to record the response from the brain. So common ways of doing this is, as Marta just discussed, TMS EEG. More typically in TMS studies, we use electromyography to record the response, but we can also use functional MRI to record that response as well. This is most common for TMS studies, but there are some cases where some of the other modalities use online stimulation as well. So this actually fits quite nicely within the existing BIDS framework. So what we have proposed to do is to use the existing events structure that exists in BIDS. So these are tabulated text files that record when stimuli happen. So typically they're related to event-related designs. So here, using these data files, we can record when the stimulations occur. We can include some additional fields related to stimulation to record how these stimulations are occurring. And then we can include some information about where the stimulation is occurring on the head. We can then use the JSON sidecars to provide some additional information about the devices themselves. So this is where we can include the what. And this is just one example in here of how we've recorded the type of TMS device that was used and the coil that was used in this fictitious experiment. The second way that brain stimulation is often used is in what's called an offline stimulation paradigm. So this is more of a pre-post condition. So here the neuroimaging modality will be used to capture the data. Then the brain stimulation will be applied, and usually in some kind of a neuromodulatory capacity, so trying to change brain function in a lasting way. and then we have a post-measurement of the brain activity. So this creates a little bit more of a challenge in the BIDS framework, because here we don't actually have a data file that's actually related to the stimulation itself. So what we've proposed is to create a new file extension for BIDS called an intervention file type. So using this file type, we can actually have a text file and a JSON file that is specific for the stimulation itself and gives us some of those parameters. Why this is useful is because we can then make use of another extension type, the scans type, to tell us when within our protocol the stimulation took place. So we've got the pre-EEG, the stimulation, and then the post-EEG. And we can then make use of the .tsv and the .json files to provide that additional information. So about how the stimulation was provided, where it was provided, and using what equipment. Another challenge is that we often use neuronavigation equipment with our brain stimulation to track where, in relation to an individual's MRI, to the underlying brain structure, that our stimulation devices are placed. So this requires additional information relating to that equipment. So here again, we can hijack either the events.json or the intervention.json to include some additional metadata related to the neuronavigation device in there as well. And importantly, we can actually include some additional information in here about which brain scan, which should also be in the BIDS format, that this particular stimulation file or this particular recording file was related to. So there are still, I would say, a number of open challenges that we are trying to work through at the moment. So one is that there are some well-established neuroimaging modalities that are captured that we use with non-invasive brain stimulation, but there are some that aren't currently in the BIDS format. So electromyography is really commonly used with TMS, but there isn't currently an agreed upon standard for EMG data. There's an extension proposal for it, but we don't have a good answer on how to share that kind of data yet. We've covered the main sort of most widely used NIBS methods with our current proposal, but there are a number of others that are currently emerging and are continuing to be developed. So we've tried to make the framework as flexible as we possibly can so that it should hopefully be able to incorporate other stimulation modalities as they become available. Something that we have really fought hard for and struggled with and gone backwards and forwards with is trying to find a balance between the breadth of advice that we give on how to capture the metadata related to stimulation, but also trying to keep it simple enough that it's usable for people. And I think we've probably gone too far in one direction and too far in the other direction at various different iterations of this proposal. And I think we're hopefully sort of sitting in a bit of a sweet spot now. But it's a real challenge to, particularly when you're working across multiple different types of stimulation that all have different parameters that are important to that type of stimulation, to capture all of that in something that's understandable has been really tricky. And I think a future challenge that we're going to have is going to be community buy-in, and this is I guess the stage where once we've put the proposal, finalised the proposal, that we actually get people in the community to use it and share their data. It's for those of you who have worked with BIDS, there is a bit of a learning curve at the start for understanding how the structure works, but then two, understanding how to wrangle your data to put it into that structure. There are converters that are available, but still learning how to use those is certainly a bit of a learning step. So I think there's going to be an education phase that's going to be really important to get this buy-in from the community. So I just want to finish off by saying a big thank you to the BIDS team in particular. They've put together a really fabulous resource, and this is again a community-run project, and they've been really helpful in putting this together. A big thank you again to all of the contributors to the extension proposal, and in particular to Giacomo, who's really been the one pushing this forward, and Tamara and her team for getting this up and going. It's been really great. I've just popped the URL up there for the proposal. It's at a fairly advanced stage, but we're certainly still open to feedback and comments. So if any of you are interested in going in and checking it out and popping a comment up there, or if you've got some suggestions, we'd be more than open to that. So please feel free to do that, and yeah, keep your science open. Thank you.
Thank you. Are there any questions?
I absolutely love this idea, firstly. I was just wondering, at what stage do people upload the data? Like, is it immediately as you collect, then you'd upload and format, or you play around with it and then after a little while you upload? Like, what's the vision around that?
Yeah, thanks, Jodie. So the typical way in which this is done is that it's the raw data which is shared. So it's pretty much either off of the device that it's recorded, or there may be a conversion step to get it into one of the data standard formats, but it's as close to the raw unadulterated data as you can possibly get. I think in a perfect world, you would finish your experiment, collect all of your data, and then, before maybe you've even published your results, you could put it up to Open Neuro. I think in practice, most people will typically collect their data set, convert it to BIDS, write their paper, publish the paper, and then maybe as part of the paper, we'll have that as an open data set. Or in some cases, they'll actually write a separate paper that is about the data itself. So there's, and I know some groups that have even done this retrospectively. So they've gone back to old data sets, which they have sharing agreements with, and have BIDSified their data and then shared them. So there's a whole range of times across the process where you could choose to share.
Oh, good, thank you. Hopefully we can contribute to this.
Great.
Hi, thanks for the talk. It was very fascinating. I'm very happy to see this initiative. I have a question, maybe that also relates a little bit to Marta's talk. So I think to really make BIDSification useful is if we also have like the next step, which would be like standardized processing pipelines, containerized pipelines that can deal with the BIDS standardized data. And I know you have done a lot of work also in the standardization of pre-processing. Are there any initiatives ongoing for development of those like kind of plug and play containerized processing also for TMS EEG data that would also capitalize on this BIDSification?
It's a really great point. Actually, Giacomo and I, last year, we were at a point where we were trying to figure out where to take it because we weren't quite happy with where the structure was at that we'd proposed. We thought it was too complicated. And actually, the point that we went back to was like, okay, what are we actually going to do with this data? Like, why would we need it in a machine-readable format? And the point that you made exactly is what we came to. It's like, okay, so what is the key information that we're going to need for TMS EEG data, put it into a containerized pipeline? And the other one that I think really helped us out was actually SymNIBS, so for electric field modelling. So we thought, that's probably what people would want to use this for, right, is if they've got an MRI scan, they've got their neuronavigation coordinates, you store that, you can then suck all of that information in, and we actually have been working with the guys from SymNIBS to try and make sure that we've got this in a standardised way that would be able to be taken up into their program for a standardised pipeline. So I think I envisage that there'll probably be a bit of backwards and forwards from both directions. So with one, making sure that the data is in a format that is usable, but then the second will be on the development side from the people working on the pre-processing to try and make sure that they can, then grab that data and pull it into their pipelines. But that is kind of the end goal, is to have, standardized workflows right the way across the whole pipeline.
Cool, thanks. Yeah, I would be happy to contribute to this.
Yeah, fantastic. I'll come have a chat afterwards.
Yeah,
that'd be great.
Thanks. Yeah.
Yeah.
Okay. Well, maybe about the standardization of pipeline in TMS EEG. I think we are very far from having a standardized pipeline because, well, first we don't agree on how the pipeline should be done, and also because when we get rid of some, we can get rid of some artifacts during the recording, so that makes a bit hard to just have one pipeline that fits any data set because it depends how the data set was recorded. So maybe something that we should aim at is having like a pipeline that also includes the recording phase in a way. And I really hope that this first study of the TATE initiative, the Team for the TMSCG initiative, will lead us closer to understand which practice we can just get rid of and which we can keep, so we are closer to a standardization. But for now, I think we are very far. Okay, I don't see any other question on the app, so I want to thank the speakers so much for explaining very well all the initiatives, and I want to thank you for being here. I hope this is inspiring for you, and you might contribute to some of these initiatives or others. Because, collaboration is the way we should go to really have definitive answers to how the brain works. Thank you for being here today.